Organic chemistry is the study of carbon-containing compounds. These compounds are the building blocks of life. They form the very fabric of the cells in our bodies, the food we eat, and the clothes we wear. Organic chemistry is a vast and fascinating field. It explores the structures, properties, and reactions of these compounds. In this exploration of organic chemistry, we delve into the realm of advanced concepts. We will uncover the secrets of complex structures like proteins and carbohydrates. We will journey through intricate reaction mechanisms, and we will discover how these molecules shape our world. Imagine the world of organic molecules as a vast and intricate universe. Each molecule is a star, each reaction a supernova. As we explore this universe, we begin to understand the fundamental processes that govern life and the world around us. This journey will take us through the fascinating realms of aldehydes, ketones, carboxylic acids, proteins, and carbohydrates. Fasten your seatbelts and let the exploration begin.
Section 5. Proteins, the workhorses of life. Proteins are complex macromolecules that play essential roles in all living organisms. They are involved in a vast array of biological processes. These range from catalyzing biochemical reactions to transporting molecules across cell membranes. Proteins are polymers of amino acids. Amino acids are organic compounds containing both an amino group, or NH2, and a carboxyl group, or COO. The specific sequence of amino acids in a protein determines its unique three-dimensional structure and function. Proteins exhibit 4 levels of structural organization. The primary structure refers to the linear sequence of amino acids in the polypeptide chain. The secondary structure describes the local folding patterns of the polypeptide chain, such as alpha helices and beta sheets. The tertiary structure refers to the overall three-dimensional shape of a single polypeptide chain. It is determined by interactions between amino acid side chains. The quaternary structure arises from the association of multiple polypeptide chains to form a functional protein complex. The Buret test is a common laboratory test used to detect the presence of proteins. This test relies on the reaction of peptide bonds in proteins with copper ions in an alkaline solution. The reaction produces a violet-colored complex that can be visually detected. Please, before continue this video, take a second to strike the subscribe button and like this video by Taiwo Falana. Thanks. Section 6. Carbohydrates, fueling life's adventures. Carbohydrates are a ubiquitous class of organic compounds essential for life. They serve as a primary source of energy for living organisms. They also play crucial roles in structural support and cell signaling. Carbohydrates are classified based on their size and complexity. Monosaccharides are the simplest carbohydrates. They cannot be hydrolyzed into smaller sugar units. Common examples include glucose, fructose, and galactose. Disaccharides are formed by the condensation reaction between 2 monosaccharides. Sucrose, lactose, and maltose are examples of disaccharides. Polysaccharides are complex carbohydrates composed of long chains of monosaccharides linked together. Starch, glycogen, and cellulose are examples of polysaccharides. Benedict's test and the iodine test are commonly used to detect the presence of carbohydrates. Benedict's test specifically identifies reducing sugars. These are sugars that can donate electrons to other molecules. The test relies on the reduction of copper ions in an alkaline solution. This results in a color change from blue to green, yellow, orange, or red, depending on the concentration of reducing sugars. The iodine test, on the other hand, is used to detect the presence of starch. Starch molecules form a helical structure. Iodine molecules can get trapped within this helix, resulting in a characteristic blue-black color.
Section 7. Detectives in the kitchen. Unmasking the molecules of life. Food tests are simple yet powerful tools that allow us to identify the presence of different biomolecules in food. These tests exploit the unique chemical properties of carbohydrates, proteins, and lipids to produce observable changes. The Benedict's test, as discussed earlier, is used to detect reducing sugars. A positive Benedict's test, indicated by a color change, suggests the presence of simple sugars like glucose or fructose. These sugars are abundant in fruits, honey, and processed foods. The Buret test helps us identify proteins in food. A purple solution after adding Buret reagent indicates the presence of peptide bonds. These bonds are the building blocks of proteins. Dairy products, meat, and legumes are excellent sources of protein. To detect fats, we employ the emulsion test. Fats are not soluble in water. When mixed with water, they tend to separate. However, adding a small amount of detergent breaks down the fat into smaller droplets. These droplets remain suspended in the water, forming an emulsion. A cloudy appearance after shaking a mixture of food and water with a bit of detergent indicates the presence of fats. Common sources of dietary fats include oils, butter, and nuts. These food tests provide a glimpse into the fascinating world of food chemistry. They allow us to make informed choices about our diet and understand the building blocks of the food we consume.
Section 8. Organic Chemistry, a Universe of Applications. Organic chemistry is not confined to the laboratory. It permeates every aspect of our lives. From the clothes we wear to the medicines we rely on, organic compounds play a pivotal role in our daily existence. Alkanoic acids, with their distinctive smells, are key ingredients in perfumes and flavorings. Think of the sweet aroma of vanilla or the pungent scent of vinegar. These aromas are due to the presence of specific alkanoic acids. Aldehydes too find applications in the flavor and fragrance industry. The characteristic smell of cinnamon is due to cinnamaldehyde, an aldehyde. Formaldehyde, another aldehyde, is used as a preservative in some vaccines and medical specimens. Ketones, with their excellent solvent properties, are widely used in industries ranging from paints and coatings to pharmaceuticals. Acetone, the simplest ketone, is a common solvent for nail polish remover and paint thinners. Proteins, the workhorses of life, are essential components of our diet. They are broken down into amino acids during digestion. These amino acids are then used to build and repair tissues, produce enzymes and hormones, and carry out other vital functions. Carbohydrates, our primary source of energy, are abundant in foods like grains, fruits, and vegetables. They provide the fuel for our daily activities, from walking and talking to thinking and dreaming.
Section 9. Conclusion. The ever-expanding universe of organic chemistry. As we conclude our exploration of advanced organic chemistry, we realize that this is merely the tip of the iceberg. The world of organic molecules is vast and ever-expanding. From the simplest alkanes to the most complex biomolecules, organic compounds shape our world in countless ways. They are the building blocks of life, the source of energy, and the inspiration for countless technological advancements. As we delve deeper into the intricacies of organic chemistry, we unlock new possibilities for innovation in medicine, materials science, and beyond. The potential for discovery is limitless. So embrace the wonders of organic chemistry, for it is a universe waiting to be explored.